Today I wanted to introduce you to some of the concepts in the pattern of inheritance or genetics chapter in the Openstax textbook. And in this first video we will take a look at Mendel's experiments with pea plants and see how he is really figuring out some of the rules of genetics, and take a look at how we can use a Punnet square to demonstrate what's going on with these genetic traits. And if we look at the process, what Mendel used, he had gardens of pea plants, which are actually a very convenient kind of organism to use for genetics, because you can control the parents and what's going on in terms of fertilization. And if you look at this figure, what you can see is the pea plants have both parts resembling male and female parts. The stamens, if we look in this figure over here, you can see the stamens are on the ends of the flowers right here. That's where the pollen is produced, and this is the equivalent of male sperm. And what he could do is actually go in there and physically remove the stamens from flowers, and then he could take that pollen off of the stamen, and you can see, just using a little paintbrush, if you paint it onto the carpel of different kinds of flowers out there, this is really equivalent to what bees are doing when they're pollinating these plants. You are effectively controlling who the father is for the babies that will be produced in the female plant organ that's out there, which is contained inside of the carpel there. So he could take that and he could pollinate these things, and then the little eggs inside of the carpels here, or the ovum cells, would be able to mature. And you can see in this little figure right here that we have each of these individual babies that are coming out of this process, and you could take those seeds and plant them. And the nice thing about working with plants is you don't have to worry about them running away or flying away, because they're stuck right there. And the most important thing that Mendel did is he made observations on the children that came out of these crosses that he was actually able to control by controlling the fertilization that was going on there. He took copious notes, and he started to do an analysis of these observations to see if he could determine what was going on with these traits that were being passed from one plant to another. And if you take a look at this figure, what we can see is he started out, or eventually he ended up looking at a variety of different traits, but one of the ones that we see classically is this idea of flower color. And he took purple flowers and he was able to cross them with pea plants that had white flowers. And the progeny that came out of this had all purple flowers. And the term that we use for this now in genetics is we say the purple flower color is dominant to the white flower color. That does not necessarily mean purple is better or worse. It just means in genetics, when you cross purple with white, the purple color is going to overshadow that white color for the flowers there. And what Mendel did is he established some true breeding plants. And what happened with this is, by breeding purple flowers over and over and over, he would get these lines of true breeding pea plants where they would always produce purple flowers. And he did the same thing with pea plants that had white flowers. Their characteristics did not change over generations. And Mendel did this and looked at a variety of these different pea traits, and he found that they had two forms that, again, we call dominant and recessive. For flower color, purple is dominant to white flowers. And he looked at a whole bunch of different traits on these pea plants, and you can see that from here: flower position, seed color, seed shape, pod color. And he established true breeding lines for all of these different kind of characteristics where the dominant trait overshadowed the recessive traits. And so we could use these true breeding lines to start doing his genetics experiments. And we can see a similar kind of thing. If you take your tall plants that are out there and you cross them with some dwarf plants, the first generation that comes out of there had all tall plants coming out of there. So again, this sets up that precedent that the tall pea plant is dominant over the dwarf pea plant that's out there. So let's take a look at what Mendel did that was really kind of a novel sort of experiment there. He did what we now call a monohybrid cross. He took his true breeding parents, again, purple and white flowers in this example, and he crossed them. And the first filial generation, or F1 generation, had all of those purple plants. But then what he did is he took those purple hybrids and he crossed them with each other. So he crossed that first generation with other pea plants of the first generation, and he got his second filial generation, or what's known as F2 generation. And if you look at this, he found that he had a mixture of purple and white plants come out of there. But the important thing for here is, remember, he did an analysis of his results. And what he found was that if he got a massive number, we're talking hundreds if not thousands of these crosses to give progeny, he found that about 75%, or 3 out of 4 of that population, had purple flowers, and about 25%, or 1 out of 4, ended up with plants that had white flowers. And this is one of those kinds of observations that make scientists scratch their head. How could you get these white flowers from these purple flowers when you had these true breeding lines in that first true breeding generation that was out there? So this sort of sets up this idea that something strange is going on with genetics, and that the white flower was still there. It was just hidden in that first generation, and we didn't see it because the dominant trait overshadowed that recessive trait. So how do we look at this stuff? One way of doing this is to take a Punnet square, and you can think about, in this case, I'm going to call the purple flower color P, with a big P right here, and for our white flower with the recessive allele, these guys, we will use the little p to designate them. So think about a cell in these purple plants. They've got 2 copies or 2 chromosomes that have this dominant purple color, and those cells undergo meiosis. They're going to produce haploid cells that just have a single copy of that purple color gene. And we can do the same thing for our white flower over here. Those 2 characteristics are being separated when these cells are going through meiosis to make our ovum or our pollen for the plants. But those are the cells that are going to end up in the next generation there. And so we can take these and do that cross and combine our dominant or large P right here with the small one, and we get this mixture of traits right here, 1 big and 1 little. And if you think about this process for all of these crosses that we see here with our Punnet square, all of the progeny end up with 1 big and 1 little P. And so this gives us our purple hybrids. That white flower color gene is still there, represented by our smaller case P right here, but the purple gene is able to overshadow it. And when we look at these, they have a special name in genetics, and they're called heterozygotes. This cell right here is representative of our zygote, the fertilized egg cell, and hetero again means different. So these zygotes have different versions or different alleles of our gene that ended up in them that came from their parents that are out there. And likewise, if we use this similar kind of terminology, if we're looking at our purple flowers that are out there that are true breeding, those are known as homozygous dominant. Homo for same, so they have the same dominant trait inside of their zygote. And finally, for our white flowers, they're known as homozygous recessive. They have the same gene, the same recessive gene in their zygote, and that gene results in those flowers having that white color there. This begs the question now, what happened with his 2-generation crosses? Remember, he took white and purple true breeding lines and he crossed them together and he got his purple hybrids. And what we see here is, if we use our Punnet square to depict the genetics for what was going on with that 1st-generation cross. So we're crossing our F1 children with the F1 children here. They both have purple plants, and they are both heterozygous. They have 1 large purple or dominant trait and 1 smaller case or recessive trait there. That is the one that we're looking at for our white flowers. And we take them and we run them through meiosis just like we did previously, and we can take a look at our results here. These alleles are going to separate out. we're going to have our purple allele right here, and this particular set of children have both dominant traits there. So those guys are going to be purple in color. These ones over here have the large and then the small one. So these two right here, those are the heterozygous purple hybrids that are similar to the parents there. And then you get this last case right over here. This is the one that has the two recessive copies of that gene. And so these are the ones that have that white color. And so if you look at your F2 results right here, this is how he got that 3 out of 4 purple and 1 out of 4 are white. Again, if you were just to look at 4 plants, you probably wouldn't get this perfect kind of proportion. But Mendel looked at hundreds, if not thousands, of these crosses. And the more crosses you do, the more chances or the more probability that you're going to approach this 75% of that population is purple and 25% of them are white. So when we look at this, what Mendel concluded by thinking about his results here is that plants are inheriting 2 alleles for each character that we're looking at. And what we know now is that term allele actually refers to a gene on a chromosome. And so if you think about this process here, you have 2 chromosomes in these parent cells here. They go through meiosis and we separate those alleles or chromosomes right there. And then they are recombined to make our diploid plants right there. And that's where we get our characteristics coming out in this second filial generation here. The other interesting thing, if we take a look at the types of plants that are out there, we can divide them up into 2 different ways of looking at them. One is what's known as the phenotype, and this is what we actually observe as a result of those alleles being used inside of the cells right here. And so when we look at this population of plants out there, 75% of them are going to be purple in color. But notice, there is a difference in terms of the genetics for these plants. The dominant trait is overshadowing the recessive trait in 75% of that population. But for these guys over here, the phenotype is actually coming from that recessive trait. So you're not actually seeing the effects of that purple color gene here. The other thing when we consider this idea is the other way of looking at the type of plants is what's known as genotype for these new cells. And if we take a look at that first combination in our upper left corner here, these are the homozygous dominant genotype because they both have that dominant gene or dominant allele there. You've got these 2 on the corners right here. Those are the heterozygous hybrids. Again, they have a mixture of a dominant and a recessive trait, but we see the phenotype of that dominant trait because it overshadows the recessive. And then the last class of these types of cells that we see is those homozygous recessive ones, where they have both recessive alleles in there. And so that's where the phenotype is coming from. And hopefully this is going to help you understand what's going on with the genetics part of this chapter.